Okay, folks, while people are getting comfortable and getting situated for our 2-hour all-in-one biology, I thought I would address a couple things. One thing I did notice on socials today was someone asked that this was part 2, how would they get part 1? And this aligns with TEAS. So part 1 is anatomy and physiology, part 2 is biology, part 3 is chemistry, and part 4 is scientific reasoning. And I know that my colleague Amber did part 1 anatomy and physiology the other night, and that could be found on YouTube. And you can look for the others. I'm sure those are upcoming, but just to give you an idea of what part 2 really stands for. Some other information I'd like to share with you. If you take a look at the screen that I have displayed, this is our free live webinars, and obviously you can access some other ones here, those others that I talked about, 1, 3, and 4. Also, we have over 1,500 free TEAS questions for you to customize your own question bank, for you to customize your own assessment. They have detailed solutions and rationale for why answers are not the correct answer. And that is all free here to serve you. So you want to take a look at those as well. And then we do offer on-demand videos. So tonight is a superficial all-in-one, but obviously there are a lot of topics that you might need to go into deeper, a little more closely, and those are offered as well. Finally, I wanted to draw to your attention that we do have tutoring, and all subjects are covered. Prices start at $25 there, and that is an option as well. And then if ever you want to contact us for the TEAS, it's prenursing@archerreview.com. All right, it's a long night, so we're going to go ahead and get started. So the topics that we are going to cover tonight encompasses all of the TEAS S2. I went in the order, for the most part, from small to large. So that means we will start with macromolecules. Then we will take a look at cells and microorganisms. Then we will move into genetic material and proteins. And then finally, we will wrap up with Mendel's laws of inheritance. One of the things that's so important for us to understand is to get a gist of biological hierarchy and where biology fits into that whole picture. So for levels of biological hierarchy, we really start at the smallest, most basic, that's an atom. That's something like oxygen, hydrogen, carbon. Then atoms can combine to form molecules. Dioxide, carbon dioxide, things like that. And then molecules can combine to form larger macromolecules. And macromolecules will be where we really dive in tonight. And those are structures that help build who we are. And those are structures that we also look to supply the nutrients that we need. And macromolecules actually make up us and our cells and are the reason why we're here. And so we will see that the macromolecules, carbohydrates, lipids, nucleic acids, and proteins work together then to form the organelles that are in our cells. And if you drop the ELLE off of there on the end, you can kind of think of an organelle as an organ that's inside our cell. And these are things like the nucleus, ribosomes, mitochondria, lysosomes. Then when we think in terms of cells, we have kind of two ways that we can look at that. We can look at the cells that then further make up the living organism, but we can also look at cells that make up singular celled organisms. Those are actually the most abundant on this planet. And we will take a look at both single-celled organisms tonight and the cells that make up multicellular organisms. And as you know, humans are multicellular organisms, and we actually have more than 250 types of cells. And we'll look at tonight how we get to those 250 types of cells. So when we continue thinking along our small to large level of biological hierarchy, cells will work together then to form tissues. like epithelial tissue, connective tissue, and so on. And then similarly functioning tissues will work together to form our organs. Like our heart is made of cardiac muscle tissue, our brain is made of nervous tissue. And then similarly functioning organs will work together to make their organ systems. Like the digestive system has organs that are focused on digesting and utilizing the nutrients in what we eat. And then finally, the organ systems all work together to support the fully functioning organism. And what's important for us to understand is that chemistry really covers that top part. And anatomy and physiology covers that bottom part. Our focus is right here. And we're going to start right there. So our learning objectives with the macromolecules are we want to understand how macromolecules are built. That's the fancy word for it, synthesized. Anytime you see that in biology, that's a word that biologists love. Just think build or make. So this is saying we want to recognize how macromolecules are built. We need to look at how they are catabolized or broken down. And we need to understand their structure and their function. So macromolecules are polymers made of monomers, and let those prefixes really help you out. Mono means one. Poly means many. So macromolecules are molecules made of many monomers. That's really all it boils down to. And macromolecules make up us, as I said earlier, and they are also broken down from our food, like this apple here, and converted into ATP, that's the structure here, which is our body's fuel. So macromolecules are incredibly important for us. And as I said when I introduced our learning objectives, we are going to focus on how macromolecules are made, that's the synthesis, and how they are broken down. Because they're made in order to support our body structures and inside of us, and they're broken down in order to help supply the energy that we need. And so they undergo two different chemical reactions, and the name of these chemical reactions with the illustrations I have here will help us make sense of them. So the first chemical reaction, called a dehydration synthesis, is where we're building the macromolecule. So remember, synthesis means to make. Now let's think about what we know about this word here, dehydration. Hydration, hydra, water. D generally means removing or in opposition of, things like that. So if we use the name of this chemical reaction with our visuals, we can determine that we're building or putting together our macromolecule by removing water. And so what this means is that in order to link our monomers together, we undergo a chemical reaction that removes a water molecule and binds them together. And that's how macromolecules are synthesized. Now, on the flip side, if we use our visuals and our word here, hydro, water, lysis means to break. To break, there's our water. In this case, if we need to break down a macromolecule, if we need to catabolize it, we are going to undergo a chemical reaction where we add water to break it. And that is how macromolecules are either built or broken down in our bodies. We talked about macromolecules briefly when I introduced the levels of hierarchy. There are four main classifications: carbohydrates, lipids, nucleic acids, and proteins. And we're going to take a look at all of those. Carbohydrates monomer is known as a monosaccharide. Looks like this. When they are synthesized into a polysaccharide, they look like this. An example of a polysaccharide is an Oligosaccharide, each of these structures on the outside of the red blood cell are oligosaccharides, and we're going to take a look at their function next. So carbohydrates come in many different forms: monosaccharides, disaccharides, polysaccharides. An example of a polysaccharide would be glycogen, which is stored in both our liver and our muscles until it's needed for energy, and then it's converted to glucose. And then the oligosaccharides, like I talked about earlier, and these are important for cell recognition. So if you notice on the top of that cell membrane, there is a label, a glycoprotein, and then there's another label, glycolipid. The glyco in that is our carbohydrate, and it's attached to, in the case of a glycoprotein, a protein molecule. In the case of a glycolipid, a lipid molecule. And what those glycoproteins and glycolipids do is they let the immune system know that that is a cell that belongs to you so that the immune system doesn't attack. So they are used in cell recognition. Lipids monomer is fatty acid. Its polymer that we're going to focus on is the lipid bilayer. And an example then would be the lipid bilayer of the cell membrane. And so we're going to shine the spotlight. There are many different lipids, but we're going to shine the spotlight on the lipid bilayer. That is one that you'll see time and time again and is important to the TEAS. And so we will certainly focus on that first. So the lipid bilayer is a double layer of phospholipids. And in this situation, any cells have a watery environment inside and a watery environment outside. And this cell membrane is a lipid bilayer. That's what they call this. And what's unique about this structure is that phospholipids have parts that are not fond of water and they have parts that are fond of water. And so they orient to support that. So if we take a look, our illustration will pop up here. I'm noticing on restreaming that I was talking a little faster than it's popping up. So I'm going to try to slow down so that what I'm talking about is actually on your screen. So there is this region here in the center that is considered a hydrophobic region. It's a region of the phospholipids that do not respond well to water. You can let the word help you out there. Hydrophobic. We know if we have a phobia, we're afraid of something. So you can kind of say that this part of the phospholipid is afraid of water. And so the lipid bilayer orients to where those tails are in the center of the lipid bilayer so that they don't have to touch the water. On the flip side, then, the other part of the phospholipid, those round heads, are fond of water. They're hydrophilic, and so they orient towards the watery exterior environment or the watery interior environment. And that's what's unique about the phospholipid bilayer and about phospholipids as a whole. Next, we're going to take a look at proteins. Proteins monomer is amino acids. Amino acids are composed of 3 primary structures: an amino group, a carboxylic group, and an R side chain. And it's the R side chain that primarily determines what type of amino acid it is. There are about 20 different amino acids in our bodies. When those amino acids link in their synthesis reaction, they form a polypeptide. And that is a long chain of those monomers, those amino acids. And an example of a fully functioning protein after that polypeptide has been folded is a muscle protein. We're actually going to shine our spotlight on enzymes as proteins because enzymes are one of the most important proteins in our body, and they're also really important aspects that you'll see on the TEAS as well. So enzymes are proteins that assist chemical reactions without being consumed. So they're helpers, but they're not used up in that chemical reaction, which means that they can be used over and over. And that's something that's certainly beneficial. What they do to a chemical reaction is they lower the energy required for the reaction to occur. And they do this by locking in with their substrate. So that's what our visual is on the screen. They lock in like a key and a lock type orientation. And that's good and that's bad in the sense that if the temperature is different than that enzyme is comfortable in, or if the pH is different, it can change that shape and it can impact the enzyme's function, and it won't function as well, or it may not function as at all. Next, we are going to take a look at our final macromolecule, nucleic acids. Nucleic acids monomer is a nucleotide. Nucleotides are made of 3 components. And I'm going to try, since we're having a lag on the screen here, I'm going to try to use my pen and see how well this works for us. So our 3 components of the nucleotide is a nitrogenous base, a sugar, and a phosphate group. I'm trying to see how long that before that lag shows up on your screen. Okay, it's coming. Examples of the polymer of a nucleotide are DNA, which is pictured, RNA, which is single-stranded, and then ATP, but ATP is not illustrated here. An example of a polymer then would be a chromosome. This is where our DNA coils up tightly into that chromosome. And we will shine our spotlight next on DNA versus RNA, 2 nucleotides that are very important in many, many different cellular processes. So DNA versus RNA. These are both nucleic acids made of nucleotides. What they have in common is we will learn that they are both involved in protein synthesis, which we will talk about later, and that they are both impacted by mutations. For the most part, then, they're different. DNA is the double-stranded spiral that you see on your screen. RNA is a single-stranded half of a ladder. Both of them have bases that make up the rungs of the ladder. And with one exception, those bases are the same. So C stands for cytosine, G for guanine, and those are complementary pairs. And those are complementary in both DNA and RNA. Adenine is present in both, but where the variation lies is that it's thymine in DNA, but it's uracil in RNA. And so anytime we have RNA interacting with DNA, we will see that if we have an adenine, it's going to pair with a uracil instead of pairing with a thymine. Both of them are responsible for our genes to protein pathway, and genes determine proteins and proteins determine traits, and we will look at that more closely later. Then RNA, there are 3 types of RNA. These are all involved in protein synthesis, and we will look at that more closely later, but they are mRNA, tRNA, and rRNA. All All right, next we are going to wrap up this section with some key takeaways, and then we'll check in and see if you have, if you're able to answer a few questions about what we've learned so far. So some key takeaways. A dehydration synthesis reaction is our chemical reaction that's going to lose water to form the macromolecule. Hydrolysis, on the other hand, is the exact opposite. It is going to use water to split the macromolecule into its individual monomers. Carbohydrates come in many forms: mono, di, poly, and they play important roles in cell recognition and energy provision. Lipids, like the example we looked at, the phospholipid bilayer, create a semi-permeable barrier that regulates the movements of substance in and out of the cell. Proteins are made of monomers known as amino acids, and they play an important role, as we saw, as enzymes. And enzymes are function to support chemical reactions. Nucleic acids, then finally, are important in holding our genetic code. That's our DNA. And we will see as we look at protein synthesis that they are involved in transcribing and translating proteins. And then also ATP is a nucleic acid, and that's responsible for our energy, our body's fuel. So what we're going to take a look at next is I have a series of 3 multiple choice questions, and I'm going to read through it with you, give you about 45 seconds to answer it. And if you want to put it in the Zoom chat answer, only I see those answers, so you can get a sense for whether you were correct or not without anyone else seeing it. Now, if you're watching this on Facebook or YouTube, you don't get that anonymity, but you're welcome to put your answers in there as well. We will then go over the answers and talk about any rationale as to why some of the other answers are not correct. So let's take a look at our first one. All right, our first one's coming up. As I said, I'm keeping an eye on the restream, and there is a bit of a lag there, so I'm wanting to have a little patience here. Okay, this is a 2 fill in the blanker with 4 choices. So blank loses water to construct macromolecules, whereas blank uses water to break down macromolecules. Is it A, hydrolysis versus dehydration synthesis? B, dehydration synthesis, hydrolysis? C, lipids or nucleic acids? Or D, carbohydrates and proteins? And I've got some quick ones already giving me answers. You guys got 45 seconds, and we'll be back together, and I'll go over the answers. Ten seconds for anyone keeping count. Okay, well, it looks like those of you that did choose to respond, very well done. You all got it correct. It is B, dehydration synthesis loses water to construct that molecule, and then hydrolysis is going to use water to break down macromolecule. So very well done. A was the opposite of, so that knocks that one out of there. And then C and D were just examples of other macromolecules. So that's what made those incorrect. All right, we've got another. I'm just waiting for that lag so that we're reading it together. All right. Which of the following best describes the role of phospholipids in the cell membrane? A, they store energy for the cell. B, they create a semi-permeable barrier for the cell. C, they catalyze chemical reactions within the membrane. Or D, they provide structural support to the cell wall. Go ahead and get your answers out there, and we've got 45 seconds. Well, we have some incredibly smart attendees. Everyone is correct that it is B, it's creating that semi-permeable membrane. So phospholipids do not store energy for the cell. I will say lipids as a broad category do have some energy storage features, but that is not a phospholipid. Enzymes catalyze chemical reactions, and our cells, which we will get to, do not have a cell wall, making D incorrect. Let's take a look at our next one. When an enzyme is degraded into monomeric structural units, the monomers will be A, amino acids, B, nucleic acids, C, fatty acids, or D, nucleotides. This is a challenging one. You got 45 seconds. The challenge with this one is to remember what category an enzyme fits into. An enzyme is a protein. And so proteins monomers then are amino acids. So I could see where some of you may have said something in the B or D range. Remember that nucleotides are the monomers for nucleic acids. And fatty acids are the nucleotides for, I'm sorry, and fatty acids are the monomers for lipids. I apologize on that. Okay, we are going to move on to cells and microorganisms. That's our next category. So hopefully you're still here with me. Looks like we have about 22 people. I'm so excited for my first webinar to have that many people. So thank you for being here with me. Our levels of biological hierarchy then for cells and microorganisms will be focusing on the organelles within the cells and on the cells themselves, both unicellular and multicellular organisms. So just to give you an idea of where we're headed with this next category of our all-in-one Archer Review biology webinar tonight. Our learning objectives for this category include identifying cellular classification, determining the structure and function of the key organelles within cells, identifying microbes that are responsible for disease, and then distinguishing between the various types of microscopes. If you're noticing long pauses, like I said, I'm really just giving time for the lag. It pops up on my screen right away, but it takes a little bit on the restream. So if you're wondering why is she pausing so long between, that's why. Okay, so what we have here on the screen is a flowchart that gives us a lot of information about how cells are classified. And I'm going to do something a little extraordinary here, a little out of the usual. And I'm actually going to start with prokaryotes. I'm going to start on the right side. And that's because prokaryotes are simple primitive cells. Scientists actually believe that they are the first cells on Earth and that those are the cells that make up our cells. And so it's more logical to start with them and move to the more complex cells such as us. But as a broad overview, cells are categorized as either prokaryotes or eukaryotes. Prokaryotes are unicellular, meaning they are made of one cell. They're very simple. They don't really have what is considered true organelle, although they have ribosomes. And their DNA is not protected in a nuclear membrane like ours is. It just floats freely throughout that cell. Now, flipping over then to the left, these are the more complex cells. These are the cells that make up us, and these are the cells that make up plant cells. Eukaryotic cells are typically multicellular, but we will see examples of single cellular eukaryotes. It happens, it's just not very often. They're much more complex than prokaryotes. They have a nucleus that protects our DNA, and then they have many specific membrane-bound organelle with unique jobs, which we will look at shortly. If we break down and look at animal cell versus plant cell, our cells have no cell wall. We have a cell membrane around our cell, but we do not have a cell wall. That is something that is unique that prokaryotes and plant cells share. Prokaryotes have a cell wall, plant cells have a cell wall, but then plant cells will still have a membrane within that wall. Both animal and plant cells have mitochondria. Those are energy producing organelle. And plants are lucky enough to have chloroplasts as well, which uses the sun to convert sun energy into a usable form. Our cells have lysosomes and centrioles, whereas plant cells do not. And then our cells have lots of small vacuoles, and plant cells have one large vacuole for water storage and other storage items. Now, at this point, because you're studying for the TEAS, our primary focus are our cells. I see a question here. Thank you so much. What about lysosome in plant cells? No, they don't typically have lysosomes. So great question. Thank you for asking. So because you're studying for the TEAS, because your preparation is for the TEAS, our primary focus is going to be on animal cells and their organelle, and that's where we're headed next. So we will shine the spotlight on eukaryotic animal cells. Those are the cells that make up us. And we are going to take a deep dive now into their key organelle. As we mentioned earlier, the plasma membrane is the barrier that encloses the cell, and it's what is often referred to as selectively permeable. I think of it kind of like a bouncer at a fancy club that only chooses what people are permitted in this club. It determines what molecules can enter and exit the cell. And it's also just responsible for separating the internal and the external environment. The next component that we're going to focus on is the cytoplasm. The cytoplasm is a jelly-like water-based substance where the organelles are held in place. It provides structural support for the cell, and it's also a site for chemical reactions. Our next one that we're going to take a look at is the Golgi apparatus. The interesting thing about the Golgi apparatus is if you've ever noticed, it's always capitalized while most of the other organelles are not. And that is because it was named after Mr. Golgi, who discovered it. The Golgi apparatus's function is to process proteins and lipids into vesicles for transportation, typically outside of the cell. Our next one that we are going to focus on is lysosome. Lysosome is an organelle that is responsible for breaking down waste in the cell and denatured proteins. So that term denatured protein, you might be thinking, what's that? Well, denatured protein is when an example would be the enzyme that we talked about earlier. If it was exposed to a pH out of its realm, or if it was exposed to a temperature out of its realm, it would denature, it would break down. And so lysosomes would be responsible for getting rid of those broken down enzymes. And you are most welcome, the person who asked about the lysosomes. All right. So the next one that we want to talk about is mitochondria. Mitochondria, you probably remember your high school biology teacher saying it was the powerhouse of the cell. It is the organelle that is responsible for converting those macromolecules that we talked about earlier into our body's usable form of fuel, which is ATP. Next, we're going to talk about the nucleus. The nucleus is one of the things that makes our cell so unique from a prokaryotic cell because the nucleus has that nuclear membrane that protects our DNA. Our DNA is locked inside there, highly protected. So it's holding that genetic material, and the nucleus is also responsible for just managing and controlling the actions of the cell. Now, next, we're going to focus on the nucleolus, that center component. So I want you to look closely before I switch at how nucleus is spelled. Nucleus. Notice for the nucleolus that it's got that word Leo in the center of it. And that is one way for you to distinguish the difference between nucleus and nucleolus. The one that has Leo in the center of it is referring to the center organelle inside the nucleus that's responsible for producing and assembling ribosomes. And ribosomes are critical for protein synthesis. The next one that we're going to take a look at is the smooth endoplasmic reticulum. This is often referred to as smooth ER. It is responsible for making lipids and hormones, and it also helps to detoxify any drugs that might be in our systems or any toxins that might be in our system. Finally, we're going to talk about the rough endoplasmic reticulum. It versus the smooth endoplasmic reticulum, the only difference between it is that it has ribosomes peppered on its surface, and that makes it look rough, and that's how it got its name. And the rough endoplasmic reticulum is responsible for really folding and shaping proteins, and it works with the ribosomes, which we will see in a little while, are responsible for protein synthesis. Next, we're going to look at vacuoles. Vacuoles for our cells, instead of just plant cells having one large one, we have many vacuoles. And our vacuoles are used for storage, essentially. They store toxins, they store carbs, they store fluids, and they can also help get rid of some of those substances that should not be in our cells. So hopefully you're all hanging in there well with me. We're doing a bit of a transition in the middle of this one. We're going to switch to looking at microorganisms. And our focus on microorganisms is on pathogenic microorganisms. These are the ones that cause disease. So if you see that word pathogenic, you want to think disease. And microorganisms can be single cellular and they can be multicellular, but the key idea is that they are microscopic. And we'll look at a few that fit into families where the larger groups in the family aren't microscopic, but as a whole, they are. So there are 5 different pathogenic microbes that we're going to focus on, 5 different classifications. There are microscopic animals, protozoans or protists, fungi, bacteria, and viruses. Oh, and I see I've got another question here. I love it. What's the difference between the smooth and rough endoplasmic reticulum function? So the endoplasmic reticulum, the rough one, is really involved in proteins. That one's involved in protein packaging and protein production, whereas the other one is focused with lipids and hormone and that type of production. So thank you for that question. I appreciate that very much. Okay, so back to our microbes. We are going to take a look now at the different types of microbes a little bit more closely. So the first microscopic microbe that I wanted to focus on is helminths. These are our parasitic worms, things like tapeworms or roundworms, and they can live in the intestines. They can cause malnutrition. They can cause an obstruction in the intestines. An interesting point about tapeworms and roundworms, if you have pets, which I have two big Great Danes, so I know all about this, and I'm sure many of you have pets as well. When the vet asks you to bring in a sample of their feces, these are the kind of things they're looking for. So they will actually put their feces under the microscope and look for those types of organisms to ensure that they aren't infected and could have an obstruction or malnutrition. So more than you ever wanted to know, I'm sure, about parasitic animals. Our next one are protozoans or protists. Those are synonyms. These are single-celled eukaryotes. So as I indicated earlier, on occasion we can find them. It's not very often, but on occasion we can find them. Believe it or not, protozoans are the largest group of organisms on the planet. And that's mostly because a lot of them live in the ocean, and that's what makes them the largest group of organisms. From a parasitic standpoint, they can cause diseases such as malaria. And I want to be very clear about why there's a mosquito on our screen. Mosquitoes are not microscopic organisms, but they carry those microscopic organisms, such as what's in the picture above it. And when a mosquito bites you, they inject a little bit of an like a numbing agent so that you don't feel them biting you as much. And in that numbing agent, they can pass the protist, and that is what then finishes its life cycle and causes malaria. So please don't assume that a mosquito is a microscopic organism. Definitely not. Our next one we want to talk about is fungi. This category, as I said earlier, there were some that wouldn't be microscopic in all capacities, and fungi fits that. We know we can see some molds. We know we can see mushrooms. We know we can see some of those things. But the ones that are microscopic are act as parasites to us, and they cause diseases such as athlete's foot. And notice that bottom one, ringworm. Notice that weird name. Ringworm is not a worm. Ringworm is a fungal infection. It got its name because, as pictured, that skin picture there, it looks kind of like a round worm under the skin. And so it got its name, but it's a very poor name for the type of infection it is. So please don't mistake ringworm for an actual worm. It is not. It is a fungal infection. All right, I see some more questions. Thank you. Do mosquitoes die after they bite us? You know, I don't know. I don't think they do. I know that there are some bees that die after they sting us, but I'm thinking that mosquitoes live after they bite us. But I can't say that I'm 100% certain about that. Great question. I'm looking at our next microscopic organism, that's bacteria. Bacteria is prokaryotic. So remember, this is a very basic cell structure. And what's important for you, oh, you're welcome. What's important for you as a future nurse is to know that bacteria can be classified by the type of cell wall they have. If they have a thick cell wall, when they stain them and put them under the microscope, they will be that purple image on the screen. If they have a thin cell wall, that's a gram negative, they will be stained and they'll look pink instead of purple. And why that's important is because most antibiotics target the cell wall. And so that's an important aspect for you to know as a future nurse. Now, one thing that's important to understand about bacteria is not all of them are pathogenic. Not all of them are bad. Lactobacillus, what you find in yogurt, is actually very good bacteria that helps balance out the bacteria that lives in your digestive system. So it is important to understand that. But there are pathogenic bacteria that cause things like food poisoning, meningitis, which is an infection in the layers that protect your brain, or sepsis, which is blood poisoning. And finally, we're going to talk about viruses. Viruses, though they loosely put them in the microorganism category, because where else would we put them? They're actually not truly, by scientific definition, alive per se, living per se. That's why they give them this phrase, non-cellular entities, because they cannot reproduce on their own. And reproduce, or reproduction, I should say, is an essential component of life. And so because they cannot reproduce on their own, they don't really fulfill all of the characteristics of life. What they actually do is they attach to our cells and they inject their DNA or their RNA into our cell, hijacking our cell and forcing our cell to make more viral particles. So that is one aspect of viruses that fascinates me, and I find it entirely creepy as well. Viruses are going to cause diseases such as the common cold, the annual flu, measles, COVID-19. And I see some questions here. Which microorganism is microscopic? So all of them fall under the microscopic category, though they do have some classifications within the grouping that are maybe not microscopic, like fungi. We can see mushrooms. Great questions. So someone's saying that bacteria can reproduce on their own without any help. Yes, bacteria can, but not viruses. So that's a great, if I missaid bacteria at some point, that's my bad. And if someone said that, if someone caught that, thank you. But viruses cannot reproduce on their own. And there's definitely a strong distinction between viruses and bacteria, as we've seen so far. Okay, we are going to continue on here. And our next focus is the two different types of microscopes. So microscopes were invented by Anton van Leeuwenhoek in the 1600s. There are two primary types, light microscope and electron microscope. And we are going to first talk about light microscopes. So light microscopes, as the name infers, or as implies, would be a better word there, require a light source. They have a maximum magnification of about 1,000 to 2,000. In high school, you probably worked with one that had about a 400 magnification. There are multiple types, three that I just wanted to focus on. There is a bright field and a dark field, and those have to do with whatever color specimen you're looking at. So if you have a dark specimen, you would put it in a bright field microscope that has a light background. If you have a light specimen, you would put it in a dark field microscope with a dark background to better illuminate your specimen. And then there's also a fluorescent microscope where fluorescent molecules can illuminate specific structures to really draw them to attention. The next microscope that we want to talk about is an electron microscope. Electron microscopes are really cool because they use electrons instead of light for visualization. And so that helps them create incredibly vivid images. As we can see in the center of that screen is a pollen grains. So electron microscopes magnify 150,000 times the size of the specimen. So that's their max magnification, which is pretty amazing. They have incredible resolution, so you can see every little detail. But the big drawback is you can't put a live specimen under that microscope. Okay, so we are coming to the close of this particular segment, and we're going to focus on our key takeaways, and then we'll have a few questions to see what we've learned so far. So to wrap this up, we want to remember that cells are classified as prokaryotic or eukaryotic based on their complexity. And our focus, especially because you're preparing for the TEAS exam, is on eukaryotic animal cells. And they house key organelles such as the Golgi apparatus for protein and lipid processing, mitochondria for energy transformation, nucleus to manage cellular functions and to house our DNA, nucleolus to synthesize the ribosomes, smooth ER for detoxification, and rough ER for protein processing. And then the infectious microbes that we went over are things like microscopic animals are parasitic worms, protozoans or protists are single-celled eukaryotes, fungi, parasitic, and that would be the parasitic version would be ringworm. Bacteria are prokaryotic and they are assessed as gram-positive by having a thick wall or gram-negative as having a thin wall. And then viruses are non-cellular entities. They cannot reproduce on their own. So those kind of fall in the microbe category, but they're odd that way. And then there are two broad categories of microscopes: light microscope, which has a maximum magnification of 1,000 to 2,000. And then it's cut off on the screen there, but the bottom one is the electron microscope, and its maximum magnification is 150,000. All right, so I've got a question here. Dark and light microscope, what's the difference? Dark microscope, maybe you were talking about like the dark field, and that had to do with if you had a light specimen, you could have a dark background. So I hope that's what you were asking. All right, we're going to take a look at a check and see what we know so far. So this is a fun one where we're going to do some matching. So we have our structures on the left and we have our functions on the right. And so I'm going to give us about our usual 45 seconds, and then I'll reveal and you can check yourself. So this isn't one where we really get the opportunity to put anything in the chat, but we can just kind of think to ourselves what we believe the correct answers are, and then I will reveal them in 45 seconds. Okay, so our first one, ribosomes make proteins. Our second one, rough ER, process proteins. Our third one, nucleus, controls the cell. Our fourth one, vacuole, waste storage. And our final one, smooth ER, makes lipids and hormones. I'll let those, I see the lag on the screen, I'll let those get caught up and then we'll move to our next one. Our next one is a fun one where you can put some answers in the chat box on Zoom or in your chat box on Facebook or YouTube. Remember, in Zoom, it is anonymous. In Facebook and YouTube, it is not. All right, our next question asks, ringworm is actually A, fungus, B, protist, C, virus, D, microscopic animal. You've got 45 seconds. I'm seeing some answers already. You guys are fast. Okay, most of you got this. It is a fungus. I saw a few guesses of protists, which I said they were the most abundant organisms on the planet, so I could see why that was a good guess. But no, remember we said this is a fungal infection that just happens to look like a worm under the surface of the skin, and that's how it got its weird name. Viruses, remember, are non-living entities, and microscopic animal. I actually was surprised no one chose that because that is like roundworm and tapeworm. And so with this worm name in there, I wouldn't have been surprised on that one. But most people did not go with that. So well done. All right, we're going to do another. Which of the following statements about prokaryotic bacteria is true? A, gram-negative bacteria have a thick cell wall. B, all prokaryotic bacteria are pathogenic. C, many antibiotics target the bacterial cell wall. Or D, pathogenic bacteria are harmless to humans. You've got 45 seconds. I'm getting lots of choices here. Well done. I'm so pleased. And we now have even more attendees. So thank you for those of you that are showing up tonight. I am loving this for my first webinar. Thank you for being here. All right. So for those of you that chose C, you are correct. So let's kind of break down why the others are not. Those of you that chose A, it's gram-positive bacteria that have the thick wall. It's gram-negative that have a thin wall. But nice, nice try. That would have been a close second. B, all prokaryotic bacteria are pathogenic. No, remember we talked about lactobacillus, which is in yogurt, and that's actually very beneficial for our digestive health. And then pathogenic bacteria are harmless to humans. No, that word pathogenic means harmful. So that makes that one incorrect. Okay, folks, how you hanging in there? You doing okay? We are moving on to a new topic. Our next topic is cellular processes. So I always like to ensure that we know where we're at in this level of biological hierarchy. So with cellular processes, our focus is going to be on the organelles within the cells and then the cells themselves. So we will start off with the organelles and go from there. Our learning objectives for this section is to identify the key structures and functions in DNA replication and cellular division. We will review the relationship between the nucleus, chromosomes, genes, and DNA. We will determine the difference between mitosis and meiosis. You might see some words on there that are a little unfamiliar. Somatic cells are body cells, and that's what mitosis divides. Whereas gametic cells are sex cells, and that's what meiosis divides. So we're going to take a look at that. Someone's thanking me for discussing the topic, so you're welcome. I appreciate that. And then finally, we're going to look at how do cells become specialized. We just looked at all the different organelles that cells have, but the truth is some cells don't have those organelles. And so we want to kind of consider, well, how does that happen? What happens there? And that's how we will wrap up this category. So what's interesting to think about with a cell is that a cell has a life cycle, much like we do. You know, we have newborn, we have toddler, teenager, adolescent, senior citizen, so on. And a cell has phases or stages the same. They have interphase, and during interphase, they have 3 different stages. They have where G1, where the cell's undergoing normal cell functions. Then they have what's referred to as the S phase, where DNA is doubling in a process known as DNA replication. And then we have G2, which is where it's preparing for that division process. And that division process in all the body cells, not the sex cells, but the body cells, is mitosis. And in mitosis, the focus of that is really to separate the DNA so that then in cytokinesis, we can split into 2 new daughter cells. So our first thing that we want to focus on in this process is DNA replication. How our cells double DNA in order to make these 2 new genetically identical cells. In order to focus on DNA replication, we really have to review a few things about DNA so that we know that we're all on the same page. So as we've talked about tonight, DNA resides inside the nucleus. It's in the shape of a double helix. That's the structure all the way on the right of your screen. And the sides of that double helix are the sugar and the phosphate of the nucleotide. If you think back to when we first started talking about our macromolecules tonight, we had a nucleotide with 3 parts to it: a sugar, a phosphate, and a base. And the sugar and the phosphate make the sides of the ladder. It's the base that meets up with another base, its complement, to make the steps or the rungs of the ladder. And this is where that adenine pairs with thymine to make the step of the ladder, or guanine pairs with cytosine to make the step of the ladder. And those complementary bases are bound together by hydrogen bonds. And hydrogen bonds, if you remember from chemistry class, are very weak bonds. And that's important for DNA replication. So DNA replication, we're going to look at in a 2-step process. The first step involves an enzyme. Remember, enzymes are our proteins. And the enzyme is called DNA helicase. And what DNA helicase does is it acts like a zipper pull, and it unwinds the DNA and separates the bases. So it's separating the A from the T and the C from the G by breaking those weak hydrogen bonds. Then in step 2, now that we have the split open DNA, what can happen is that we can start building a new DNA strand on each old strand. And so what that means is that our new DNA is going to have 1 fresh strand and 1 old strand. And it's going to be these matching bases, these complementary bases of A's to T's and C's to G's. And this ensures that when we have our 2 new daughter cells, when we go through mitosis, each daughter cell is going to get the exact same genetic information as the parent cell. So this process, as I said earlier when I introduced this topic, occurs during one of the cell's life stages, or one of the cell's life phases. It occurs during the S phase. So once the DNA has been doubled, the cell's going to further prepare for mitosis in the G2 phase, and then it's ready for mitosis. And that's our next topic. So let's take a look. So one of the things I've often had students ask me is, why do our cells need to divide? And so that's what this screen is about. And I do see I have some questions in Zoom. Let me see what's going on here. The replication, the question asks if the replication happens in the S phase. Yes, the replication happens in the S phase. So there's no separation as far as the DNA being separated beyond the whole process. It's unzipped, the replication occurs, and now we have kind of double DNA waiting for that division process. So why do we need to divide? So we divide primarily for 2 reasons. First of all, that's how we grow, how we get from that infant to a full-grown human. And secondly, because cells do not live forever. As we can see in our chart here, our stomach cells only live 2 days. Our red blood cells live 4 months. Okay, so I got a question here. Someone asked, they're a bit confused. What is the S phase? So let's just take a look at the visual here for just a quick second. So I'm going to pull this visual back up on the screen because we certainly don't want anybody confused. So I'm just waiting for that lag and then we'll talk about it. So the S phase, if you look at your screen here, we have multiple phases in the life of a cell. So in some ways, you could kind of think of it as maybe the elementary phase of the cell, like being an elementary student. And it's during that phase, the S phase, that DNA replication is occurring. Okay. Hopefully that helped answer that question. All right, so we said that after the DNA doubled, the cell was then going into the G2 phase where it was further preparing for mitosis. And now let's take a look at mitosis. So mitosis is essentially just splitting up that DNA. That's really the bottom line. We've got doubled the DNA. We need 2 new daughter cells with the right amount of DNA in it. And so mitosis is all about splitting that DNA into 2 new cells. We've got 4 phases of mitosis. Sometimes tests can ask like the order of the phases. So I know that sometimes students will come up with PMAT as, and you're welcome, PMAT as a kind of little acronym to stand for the phases in their order. Whatever you come up with, that's the order, and I know a lot of times tests will ask about that. So the first phase of mitosis is prophase. And this is where our chromosomes are going to be fully visible. So one thing that may not be apparent, not something we really talk about much, is that DNA really exists in our cells as chromatin, just kind of loose DNA within that nucleus. And so it's not until the cell is going through preparations to divide does it coil up tightly into chromosomes. And so in the prophase aspect of mitosis, these chromosomes are condensed and they consist of 2 identical sister chromatids joined at the center with a centromere. That's that little round thing in the center. And so someone asked chromatin versus chromosome difference. Great question. So chromatin is DNA that I would say is lounging. It's all spread out within the nucleus. It's not condensed tightly. And then when that cell is getting ready to divide, it condenses up really tightly into the chromosome, which is the visual you see on the screen, the purples and the tan. And that's a chromosome. Great question. So in this phase, we've got these visible chromosomes, we've got sister chromatids, and we've got a centromere in the center. The next phase is metaphase. And metaphase is the one, if you've looked at anything under a microscope in high school, you probably were able to see the chromosomes lining up along this middle phase. And they're lining up equal from the 2 poles or the opposite ends of the cell. And the reason they're doing that is really in preparation for anaphase. In anaphase, then, the sister chromatids, which are actually being pulled apart. They're being pulled to the opposite poles of the cell because remember, our whole goal here is to split that DNA apart, okay? And so in anaphase, those chromatids are being separated. Okay, I'm seeing some more questions here. So a sister chromatid is just one leg or one arm of the chromosome. I hope that answered the question. So what we see here is these were one chromosome with 2 sister chromatids, 2 legs, and now in anaphase, they're being pulled apart. Finally, in the last stage of mitosis, we have telophase. And telophase is where we have almost 2 new cells. They're not fully separated yet, but we have almost 2 new cells. chromosomes are going to start to relax back into chromatin. We're going to get our nuclear membrane around the chromosomes, and all of that's going to start preparing it to be two separate cells. And that's really where mitosis ends. Finally, a process known as cytokinesis separates those two cells into two separate cells. And so our end result of cell division is two genetically identical daughter cells containing the same number of chromosomes as the original parent cell, and they're identical to the original parent cell. And these are somatic cell division. This is the body cells. Everything but sex cells undergo mitosis. Okay. All right, we're moving right along. Our next topic is meiosis. So meiosis is going to be the type of cell division for our sex cells. So one of the questions that I often get asked is, initially at least, is why? Why does meiosis, why do we have to have a separate division for our sex cells? And it really boils down to, we don't want to be identical to our siblings, and we wouldn't have the right number of chromosomes. So meiosis is going to divide sperm cells, spermatocytes, or oocytes, which are our egg cells. And what meiosis offers that's different than mitosis is something known as crossing over, where the chromosomes exchange genetic material so that we have genetically diverse cells at the end. That's what makes you different than your sister or your brother or all of your other sisters, whatever ones you have. And so meiosis is really important for that genetic shuffling, so to speak, so that you don't have the exact same genes as your siblings. The other thing that is important here is that mitosis gives us 46 chromosomes and meiosis gives us 23. And I want us to think for a minute, why would we need half? And I see I have another question. So the oocytes are egg cells. So the question was, what does that mean? So spermatocytes are sperm cells and oocytes are the egg cells. They are the originating cell that undergoes division in order to create a sperm cell or in order to create an egg cell. Great questions. So I ended on, why would we need half? We need half the number of chromosomes so that when the sperm meets the egg and fertilizes it, we have a full 46 number of chromosomes. If you recall, a full human has 23 pairs or 46 total chromosomes. And so if we didn't have the smaller number of chromosomes to start with when the sperm and the egg met, we'd have too many chromosomes and we have to only have 46. And so that's the whole point of meiosis, is to generate genetic diversity and get us to that smaller number of chromosomes. So in order for that to happen, meiosis actually is a process of two divisions. And in some ways, you can almost perceive it as mitosis happening twice. We have our prophase 1, metaphase 1, anaphase 1, telophase 1. They all look very similar to mitosis. Then we have prophase 2, metaphase 2, anaphase 2, telophase 2. Again, looking relatively similar. The distinctions that I want to draw here is, if you look very closely, you'll see that some of the purple chromosomes now have orange and some of the orange chromosomes now have purple. And that's showing that crossing over or that genetic exchange that leads to genetic diversity. Also notice that if we stop at the first division, we still have 46 total chromosomes, and we've got to get to 23. And so the whole point of that second division is to get to 23 chromosomes. So I see another question, the confusion about the difference between the two divisions of meiosis. So again, the whole idea of meiosis is to divide twice to get to 23 chromosomes, so that when the sperm meets the egg, we end up having the right number of chromosomes. Otherwise, the processes, as you can see on the screen, are very, very similar. The only other difference is, as you can see on the screen, they do that genetic shuffling, what is known as crossing over or recombination. And that way, when we end, we have some of those chromosomes with a little bit of orange on them that were purple, and the purple ones have a little bit of orange on them. And so that ends with ultimately 4 genetically diverse cells. It's not showing meiosis cytokinesis here, but if you can imagine a line down between them, we would have 4 genetically diverse cells. Oops. So I think a good compare and contrast are important at this point so that we understand where we're at in reference to mitosis and meiosis. So what they have in common, they are both forms of cell division. They both involve chromosomes, and they both require DNA replication in order for the division to occur. Mitosis is starting with a parent cell of 46 chromosome and ending with 2 genetically identical daughter cells with 46 chromosomes. Whereas meiosis is starting with that parent cell, the spermatocyte, and ending with our 4 genetically varied cells of 23 chromosomes. Mitosis is somatic cells. Somatic is body cells. Meiosis is germ cells. Germ cells in this context are sex cells. Mitosis, genetically identical. Meiosis, genetically unique. Mitosis, 1 division. Meiosis, 2 divisions. Okay, so I got some questions here. Does color matter, or is it just for demonstration? Yeah, color doesn't matter. They're not purple and orange, just for demonstration. Great question. Meiosis starts with 46, then after division, it has 23 at second meiosis. Yes. So that's a person asking about the number of chromosomes. Yes, if we look at our visual on the screen, when we end in mitosis, we have the right number of chromosomes for those body cells to function as body cells. When we end at meiosis, we have half the number of chromosomes we need so that whether that's a sperm cell or an egg cell, when they unite, they will then have 23 plus 23. They will have 46 chromosomes to make a functioning human being. So one of the things that my students have often asked me in the past is like, okay, great. We go through cell division, we have our meiosis, our sperm meets the egg, but how do we really get to a being? How do we go from all of these cells that have all the exact same organelles to cells that are digestive cells or nerve cells or things like that? And that's our next little topic here. So they get to a specialized place through a process known as cell differentiation. And what this really is it's activation of genes and deactivation of other genes at certain times to direct their future. And this is how we go from having our standard stem cell that has all of those different organelles to unique cells such as nervous cells or blood cells. So we're going to wrap up this section with some key takeaways. The cell cycle encompasses DNA replication and mitosis. DNA replication is where DNA is doubled. Mitosis is a 4-step process where it divides somatic or body cells, resulting in 2 genetically identical cells. Meiosis divides sex cells through 2 divisions, resulting in 4 genetically diverse daughter cells, and those are for sex cells only. And then cell differentiation is how stem cells go from being just a normal cell to being a nerve cell or a red blood cell or something like that. Okay, time to assess what we know so far. So our first question tonight, oh, you guys are awesome. I already have people answer my question. Human cells contain 46 chromosomes. After a cell undergoes mitosis, how many chromosomes will each daughter cell have? 23, 46, 92, or 12? You got 45 seconds. Because this is mitosis, the correct answer is B. Mitosis gives us the exact same chromosome number as the parent cell. Very nice job for those of you that got that. If it was meiosis, it would be A, for those of you that accidentally chose A. Let's do another. So I have a question here. During the mitosis process, does it only focus on DNA and meiosis focus on sex cells? So the correct answer there is mitosis is every other cell type in your body except for sex cells, where meiosis is only sex cells. Hopefully that helps. All right, this one we got here. What is the primary purpose of meiosis? A, to produce genetically identical cells for growth and repair. B, to create 4 genetically diverse daughter cells for sexual reproduction. C, to double the number of chromosomes in a cell. Or D, to repair damaged tissues and replace dead cells. You guys are answering already. You got 45 seconds. All right, for those of you that chose B, you are correct. They are creating 4 genetically diverse daughter cells for sexual reproduction. A and C are mitosis, and D is just nonsense. That is not a rule of either one of those processes. I do see on Facebook where someone's asking if we have handouts that you can download. And I do know that those of you that signed up for it via Zoom, we have your contact information. And so, yes, I will definitely have handouts for that. Regarding those of you on Facebook or YouTube, being this as my first webinar, that is a question I'm not certain. And so at some point when we're working on one of these questions, I will reach out to my contact and answer that for you. Another person's asking if I will discuss transcription and translation in another webinar, and honestly, that's coming up. So stay with me. All right, we have another question, and this one's a select all that apply. So in what ways are mitosis and meiosis similar? And I think this is a hard one. It says A, both involve the division of a single parent cell. B, both result in genetically identical daughter cells. C, both are processes of nuclear division. Or D, both occur in somatic body cells. You've got 45 seconds. Okay, so this one I think was a challenging one. This one is both A and C, and let me explain that. So whether it's mitosis or whether it's meiosis, they're starting with a single parent cell and ending either in 2 in mitosis or 4 in meiosis. Now, C, when they talk about nuclear division, I think that's really confusing. And what they mean by that is that you're dividing the nucleus's material, you're dividing the DNA. And I wish it was stated a little differently. But we do try to align with similar TEAS type questions. And so I certainly want you to be exposed to something like this. B, no, because they're not genetically identical in meiosis. And D, no, because that's just body cells, not sex cells. Yeah, nucleus division is another word for nuclear division, or another way to say that. And that's what someone put in the chat, and they're right. That's another way to look at that. So well done. Someone else thought A was incorrect because of being a parent cell. And we do start with a parent cell, whether it is the oocyte, which is the parent cell for an egg, or the spermatocyte, which is the parent cell for sperm. So I understand why that one would be confusing as well. And that's what we're here for, to clear up some of that confusion. All right, I hope you're still with me. We are moving on to genetic material and protein, but I do have one more question. The person asks, each with 46 chromosomes. So mitosis would be 46 chromosomes because that's making our body cells. But meiosis would be our 23 chromosomes to go into the sperm or the egg, so that when they unite to make a zygote, then we would have the right number. All right, next category, genetic material and proteins. So just touching base on where we are at on our levels of biological hierarchy. In genetic materials and protein, we are in the realm of macromolecules, as well as organelle, and that's where we're headed next. So our learning objectives for this category is to identify the relationship between genes and proteins. And we need to recognize that the genetic code is what's directing how proteins get built, and that influences our traits. We're also going to analyze transcription and translation, and that's all part of protein synthesis. And then we're going to briefly look at the impact of mutations on protein synthesis. And I did have another question in meiosis and mitosis. I'm not sure I fully understand. It says in meiosis and mitosis, chromatid would be 2 in chromosome. So I would recommend, I'm not sure I fully understand that question, either re-ask it, or when you see, because you're in the Zoom, so you'll definitely have access to the handout. So make sure you look closely at the visuals in that handout as well, and that might help. So moving into genetic material and proteins, one of the things that might be kind of hard to wrap your head around is that genes are segments of DNA, and DNA is our recipe book for making who we are. So in a very literal context, genes carry those instructions for making proteins, and it's the proteins that make us who we are. So they're determining traits like our eye color, our hair color, and that's all through multiple types of proteins. Structural proteins are responsible for our body's framework. Enzymes, like we've talked about quite a bit, can catalyze pigment production, which impacts our eye color. And then regulatory proteins, like I talked about with cell differentiation, help control when genes get activated or deactivated, and those all direct our traits. So then that makes this idea of protein synthesis, and remember that word synthesis means to make. So that makes this idea of protein synthesis a very important concept to understand, because that's determining who we are. And so in the case of protein synthesis, we're converting that recipe of DNA into the proteins. And we're doing this in 2 main stages, transcription and translation. Transcription is going to occur in the nucleus, and it's just mRNA making a copy of the DNA. And then translation is going to occur in the cytoplasm, on the ribosome, And that is going to translate the mRNA into amino acids to make the protein. And we're going to shine our spotlight on that a little further, but we first have to recall a little bit about RNA because RNA is very important to this process. So remember that RNA is the single-stranded version that looks very much like DNA. It has that different base, uracil instead of thymine. It has a different sugar, ribose instead of deoxyribose, and that there are 3 types of RNA: mRNA, tRNA, and rRNA. And those are all very important in protein synthesis. I see I do have another question about order, and I think they're referring to possibly meiosis and mitosis. And since we're on protein synthesis at this point, I think, again, you're going to get the handout. And so I would recommend you look that up. But if you're just talking about the order of stages, remember PMAT: prophase, metaphase, anaphase, telophase, if that's what you're asking. And I apologize, I didn't see that sooner. Oh, from gene to protein. Well, that's what we're looking at right now. So good question. All right, so transcription is our first step. And this is taking place in the nucleus. And I think what's so important for us to understand is DNA is so special that it is locked in the nucleus. It cannot leave the nucleus. And so that causes a problem because proteins are built in ribosomes, and ribosomes are not inside the nucleus. And so in order to build that protein somewhere else, we have to make a copy of the instructions. It's like making a photocopy. So essentially, messenger RNA is just a photocopy of a segment of DNA that is providing the instructions to build a protein. And so the most important thing here is if you look at our visual on the screen, you see that green strand that's being built. That is our mRNA. That's our photocopy of the DNA, if you will. And that is the adenine pairing with uracil, because it's RNA, cytosine pairing with guanine, and it's making that photocopy. And once that's done, once it's got the entire copy that it needs to make a protein, that mRNA strand can leave the nucleus, and it is going to travel out to the cytoplasm, and it's going to latch on to a ribosome. When it latches on to a ribosome, that is when translation is initiated. So our visual on the screen here, that big green blob, is our ribosome. And the strand that's kind of pink and blue and beige through it is our mRNA, our photocopy, if you will, of DNA. And it's laying open like a recipe book in your kitchen. And at that point, our RNA, which is part of the ribosomal unit, is really just kind of managing this whole process. And tRNA is going to be the ingredients of our protein. It's going to bring, I can't say it be, it's bringing the ingredients of our protein. So tRNA is really just a delivery mechanism. It's going to bring over amino acids, and the way that it makes sure that it has the right amino acids is on the bottom of the tRNA are bases: the A's, the U's, the C's, the G's. There are 3 of them. That's called an anticodon. And they're going to line up with the mRNA code. And as long as they have a complement, an A with a U, a C with a G, and maybe another G with a C, they know they're in the right spot, and they'll drop off an amino acid. And then that amino acid starts linking together to form that polypeptide chain. And that polypeptide chain is our protein. Once the entire recipe is read on mRNA, that polypeptide chain then will travel to other organelles, such as the endoplasmic reticulum, to be folded into a functional protein. And that's really it. That's the process. All right, you guys are still with me. I'm so proud. We even have more attendees now. Thank you, thank you. We're moving on now to what happens when there's a mistake. This is mutations. And mutations often can be harmless. And then in other cases, they can be very harmful. So we're going to take a look. All right, so I am seeing people asking about handouts. And like I said, at a point where we get to another check your progress questions, I will ask my superior how to get handouts to YouTube and Facebook folks. Those of you that are in Zoom and you signed up ahead of time, we have your contact information, so you'll get those handouts. But I'm just double checking how to get these from my superior. So bear with me there. I'm going to work on that when we get towards the end. And I have another question. Is there a written form of the order of transcription and translation? So the order is pretty much what we just went through. Transcription happens first and then translation. And that would be on the slides like we just went through. Oh, you guys are so good. You have so many good questions. I love how much you guys are learning. This is great. So mutations. Mutations will change the DNA sequence. And this can occur during DNA replication, way back when we were talking about mitosis and meiosis. And it can also occur during protein synthesis. And they can affect both DNA or RNA, especially, in protein synthesis. Some examples we are going to look at: deletion, insertion, substitution. And most are harmless, but some can lead to genetic disease. So let's take a look at some of these. So our first one that we're going to look at is a deletion mutation. And deletion mutations and insertion mutations are the most damaging mutation. If you look at the visual, what has happened in this case is a whole chunk of DNA has been cut from the DNA strand. And why that's a problem is because, remember, the amino acids are read in groups of 3. And so when the tRNA is bringing over that amino acid and it's all messed up because there's a big chunk of DNA missing, it messes up all of the amino acids that were programmed from the point of the mutation on. And so what happens here is we either end up with a protein that doesn't work right, or we terminate the building of the protein before it's done. And this can cause disorders like cystic fibrosis. As I said when I introduced the deletion mutation, the insertion mutation has the same kind of problem. If you notice in our visual here, they inserted CCAT in the middle of that DNA strand, and it messes up the reading of 3 from that point forward. And so again, we either end up with a non-functional protein, a messed up protein, or a protein that was stopped too soon before it was fully built. And a type of mutation that causes Huntington's is an insertion mutation. Finally, the last one that we're going to look at is the substitution mutation. This is actually one that typically may not even impact the protein production. And the reason that it doesn't always impact the protein production is because we're just substituting one letter for another. So maybe an A is substituted for a C, or in this case, a C is substituted for an A. And so this might be a silent mutation where it doesn't change the amino acid. Sometimes those 3 codes will code for there'll be lots of 3 base codes that code for the same amino acid. So it might, even though there was a substitution, code for the same amino acid. There could be a missense mutation where a different amino acid is incorporated. That could be a problem. Or it could introduce what is known as a stop codon, where it tells it to stop making the protein. And that could be a problem as well. A substitute mutation is responsible for sickle cell anemia because it alters the hemoglobin protein. So these are generally the least dangerous mutation, but they can still be a problem. All right, we are wrapping this one up with some key takeaways. Genes determine the formation of proteins, which in turn shape our traits. And that is a big biological concept. So it's important that we reiterate that here. Remember that RNA is single-stranded and has uracil instead of thymine. Remember that there are 3 types of RNA: mRNA, tRNA, and rRNA. Remember that protein synthesis includes transcription first and then translation. Transcription is where the mRNA strand is formed from the DNA template in the nucleus. Remember, that's a photocopy per se. Translation occurs in the cytoplasm on the ribosome. And this is where the mRNA recipe is read and tRNA is delivering all the amino acid ingredients, so to speak. And a mutation is a change in the DNA sequence that can disrupt gene function, and it might lead to disease. I know that's cut off at the bottom, but I know what that says. All right. Someone asked, what does genome mean? That is a great question. Genome is the broad term for our entire genetic recipe book. So the human genome is where they have mapped out the entire DNA sequence for a human being. Okay, we are on to checking to know what we know so far. So this one asks, which of the following best describes what occurs during transcription? A, DNA is directly copied into an amino acid sequence. B, RNA polymerase aids in the synthesis of a complementary mRNA strand. C, ribosomes assemble a protein by reading mRNA codons. Or D, transfer RNA delivers amino acids to the ribosome. You have 45 seconds. I'm already getting answers. Okay, so this is transcription, remember? And so the correct answer is B, transcription. That's our first step. What makes A incorrect is that DNA cannot be copied directly into an amino acid sequence because DNA is trapped in the nucleus by design, so it is protected. And then C and D relate to translation, the second step. Let's do another. What is the role of tRNA during translation? A, tRNA transcribes DNA into RNA. B, tRNA unwinds the DNA double helix. C, tRNA brings the correct amino acids to the ribosome, matching them to the mRNA codons. Or D, tRNA corrects mutations in the mRNA strand. You have 45 seconds. I'm seeing answers already. Okay, for those of you that had C, you are correct. You are correct. tRNA is that delivery driver. It is bringing the amino acids, or as my example, the recipe, it's bringing the ingredients to build that protein. A, no, tRNA does not transcribe DNA into RNA. So DNA is in the nucleus and it is transcribed by mRNA. tRNA does not unwind the DNA double helix. That is an enzyme, okay? DNA helicase. And then D, that is just silly. There are no RNA functions that correct mutations. All right, we have another. This one is focused on mutations. It asks us, which types of mutations can cause a frame shift in the genetic code? potentially altering the resulting protein. Select all that apply. You have 45 seconds. Okay, for this one, hopefully you chose B and C, insertion and deletion. Those are the ones that are either inputting a part of DNA where it doesn't belong or taking out a part of DNA where it did belong. And it changes then how the tRNA is lining up with amino acids all the way down. That's called a frame shift. It changes the genetic code all the way after that, and that can really alter the protein. Substitution, remember, was just a single nucleotide being changed, and a silent mutation is a form of a substitution where it doesn't even change the amino acid. Oh, you guys are hanging in. I'm so impressed. We are moving on to our final segment of the night. This is all genetics, the laws of inheritance. I'm so impressed. As with all of them, I do like to touch base with our levels of biological hierarchy so we know where we stand on our topic. So with the laws of inheritance, we are focused on the nucleic acids because those are our DNA and that's where our genes are. So that's one aspect that we'll be focused on tonight. And then really how those genes impact our fully functioning human being. And that's our next impact or our next topic in this final round for tonight. So thank you for hanging with me, and we're going to look at our learning objectives for this unit. So in this final segment, we're going to distinguish between genotype and phenotype and dominant versus recessive traits. We will analyze monohybrid and dihybrid crosses. We'll differentiate between Mendel's laws because he's the one responsible for what we know about genetics for the most part. And then we're going to look at different inheritance patterns that Mendel didn't consider. Those are considered non-Mendelian inheritance. So just as a quick reminder, we've talked about this quite a bit tonight, but it is important for us to remember that genes, which are segments of DNA in our chromosomes, determine our traits. And that becomes very important when we look at the laws of genetics. So for anyone unfamiliar with Mendel, he was a monk back in the 1800s, and he used pea plants to study genetics. And he has three primary laws that we will talk about tonight. This is one of the first ones. This is the law of dominance. And he indicated that our traits that we get are determined by genes. The traits that we inherit are determined by genes, and that each gene has an alternative form known as alleles. And I will talk in the next slide about how to kind of differentiate between alleles. But at this point, just understand that an allele is an alternative form of the same gene, okay? And he tells us that when an individual inherits alleles from their parents, the dominant allele is going to be shown. That's what we're going to see. They use the language expressed. And recessive alleles will only be expressed if there's not a dominant allele present. And this leads us to the vocabulary that we know in genetics. And so that's why we start with Mendel's first law. So I've got a couple questions. When will the next webinar of Biology 3 Chemistry be? That is a great question. I do not know off the top of my head. That is my colleague. I believe Jasmine will be doing that one. So check socials, keep your eyes out for that one. Gene and chromosome different. A gene is a segment of a chromosome. So a chromosome is our DNA all coiled up tightly, and then different chunks on that chromosome are genes, and they hold recipes for proteins. So you could kind of think of a gene as a particular recipe in a large recipe book. All right, getting to our genotype versus phenotype. So these all lean on the Mendel's law that we just talked about. So a genotype is going to be the specific genes that are responsible for a trait. And they're represented by pairs of alleles like big B, or big B, or little b, where the capital letter represents a different version of the same gene and a lowercase represents a different version of the same gene. Now, I think this can be confusing. The way I kind of think of it as a pair of shoes. If I have a pair of Nike shoes that I use every morning that I run in, my left shoe could be big B and my right shoe could be little b. They're both the same pair of shoes. If I had two big B's, it'd be like having two left shoes of that same type. Two little B's, like having two right shoes of that same type. They're the same pair of shoes. It's just they're slightly different. Now, genotype is going to be the genes that underlie a certain trait. And so you can let the prefix of the word kind of cue you in. Oh, genotype stands for gene, the genetic makeup. Genotype is what underlies the physical characteristic, what is known as the phenotype. So it's what's determining the trait, what's being expressed. The genotype is determining the phenotype. And the trait might be hair color, eye color, certain diseases. And in this case, you can let the pH and phenotype kind of cue you in. Oh, yes, that's the physical outcome. That's the trait that is expressed or that we seek. The next we have to look at is this whole concept of homozygous dominant, homozygous recessive, heterozygous. What's that all about? That's our next conversation. All right, homozygous dominant. This is when a person inherits the two dominant alleles. So the sperm would have had a big B in it, the egg would have had a big B in it, and so we're expressing the dominant trait. A homozygous recessive is the complete opposite. This is where the sperm is going to have a lowercase recessive letter, the egg is going to have a lowercase recessive allele, and you put them together and they're going to express the recessive trait. The heterozygous form is where a sperm has one letter, an egg has the other, and so we end up having both versions of the trait. And in Mendelian inheritance, this means that the dominant trait will be expressed. Okay, so if you see big B, little b, the visual outcome of that is going to be the dominant trait. That's the heterozygous version. And so that's why heterozygous doesn't say heterozygous dominant or heterozygous recessive, because in Mendelian inheritance, we know that the dominant trait is what's going to be expressed. Okay, so I had another question. Yes, I can link the slides. I did find out I do have a link for the slides for everybody. I can put that in Facebook, I can put that in YouTube, and I can put that in Zoom. So yes. And for other people who are wanting to know when we are concluding, we are so close. This is the shortest segment. We are very close. So just hang in there. I would say we are probably less than 15 minutes away. All right, our next law is Mendel's law of segregation. Okay, so we already know that each organism, before it's a sperm and an egg, has two alleles for a trait, like a capital F and a lowercase f, or like the letters we were using earlier, big B, or big B, little b. But we also know that when we go through meiosis, those chromosomes, everything's separating, and we're ending up with half the number of genes. And so we're ending up with one of those combinations. Either, in our example here, a capital F or a lowercase f, but we're not getting both because we're only getting half of our DNA in meiosis. So a sperm might have a capital F, an egg might have a lowercase f. And this is important so that when the sperm meets the egg, not only do we have the right number of chromosomes, but then these homologous pairs will align. The pairs that match naturally align. Okay? All right, moving on. So that means that we can actually predict possibility of how many offspring, or not, I wouldn't say how many, I would say the percentage of probability that someone might have a trait. So for example, in our example here, freckles are a dominant trait, and the absence of freckles is a recessive trait. So let's say that we have two parents that are heterozygous. They have a capital letter and a lowercase letter, and they want to know what's the probability that their children are going to have freckles. We can do that in a Punnett square. So what we do here is we put the possible alleles. So this first one that I have popping up on the screen, I know there's a delay, has a capital F. And that's supposing, I'm going to say dad's possibilities are up top. So that's supposing that the sperm from this male has a capital F. But because he's heterozygous, that's what they tell us in our practice problem, a different sperm could have a lowercase f. So we put that up top as well. I'm waiting for the lag so that you guys get this. The same thing goes for the egg then. If the female is heterozygous, then we put the two possibilities along the side for the female. Then when we pull those into the Punnett square, we literally just pull the one over from the side and the one over from the top. So we'll see in our top square, we're going to have two capital Fs. And that is one possible union of a sperm and an egg with this couple. Our next one, follow along with me, a capital F and a lowercase f. That is one possible combination of a sperm and an egg with this couple. Down on our bottom left, another possible combination looks the same. We've got a capital F and a lowercase f. And then our final possible combination and our bottom right, the two recessives. I got a couple of questions. Can you rewatch this on YouTube? Absolutely. It will be on YouTube. The F, a capital F with a lowercase f is called a heterozygous. And someone's already got my probability. You guys rock. All right, so when we look at a monohybrid cross like this, we want to know the genotype and the phenotype probability. And so that's what we're going to look at next. So in this case, then we see that our genotype is that we would have a 1 out of 4 possibility of having the homozygous dominant FF, capital Fs. We have a 2 out of 4 possibility of the heterozygous, capital F, lowercase f, and a 1 out of 4 probability of the recessive, lowercase f's together. When you look at that as far as a ratio, as someone already put so brilliantly in my chat, that would be a 3-to-1 ratio, or 3 out of 4 probability that would have freckles, or a 1 out of 4 probability of no freckles. And we can also report this in a percentage, that there is a 75% chance that their offspring will have freckles and only a 25% chance that they will not. All right, we are moving on to Mendel's third law. This is the law of independent assortment. This says that genes for different traits are inherited independently of each other. This means that one trait doesn't influence the inheritance of another trait. So for example, hair color is not going to influence eye color. So if we stick with my shoe example, it's saying that if you inherit a pair of Nikes from your best friend, it doesn't mean that you're also going to inherit her cool boots. These are saying there's two different, they're independently assorted. When when the DNA goes into the sperm or the egg, there's no rhyme or reason. They don't stay together, they split up. And so in that case, we can actually assess the probability of having two traits. And that does take things deeper than we'll go tonight, but I at least want you exposed to it. Someone asked the independent segment law, what's the short difference? And it just means that if you inherit your dad's brown hair, it doesn't mean you'll also inherit his blue eyes. It really is that simple. All right, so for dihybrid crosses, this is something that we would get in deeper in our on-demand videos, but for tonight, I want you to at least see it, okay? So for tonight, our example really aligns with Mendel, and Mendel studied pea plants. And pea plants would have a green color or a yellow color, and they would have seeds that were either round or wrinkled. And so in this case, he could actually use a 16 square Punnett square, think about that, to determine the possible combinations of alleles where we would have a dominant trait with a recessive and so on. And so if you want to know this further, this is something that would be exposing, or that you would see. I have a 6-step method to do this in the on-demand videos, but for tonight, it just doesn't fit into the window. But I can give you some guidance. And the guidance here with a dihybrid cross is if you have two parents that are heterozygous for both traits. So notice at the very, very top up here where it says capital R, lowercase r, capital Y, lowercase I. That represents one parent that's heterozygous for both traits. The X in the middle means crossed with. And then you see the other parent is capital R, lowercase r, capital Y, lowercase y. So these are parents that are heterozygous for both traits. If you have that, you're always going to have a 9:3:3:1 ratio. And someone already put that in the chat. So even if you never ever remember how to do a 16 square Punnett square, remember that ratio. And the 9 is that the offspring will have both dominant traits. The first 3 is that the offspring is going to have one dominant trait with one recessive trait. Same thing with the next 3, that we're going to have a dominant trait with a recessive trait. And then the single one in that ratio is both recessive traits. Okay, so even if you never get into the 16 square Punnett square, remember that ratio. Just completely memorize that, and that will make your life so easy when it comes to testing. All right, folks, we are moving on to the very last segment of tonight. This is the non-Mendelian inheritance. So remember that Mendel was a monk from the 1800s. He didn't know anything about DNA. He didn't know so much, but yet he was able to predict a big chunk of our inheritance. But what really boils down to is that so many traits are actually not inherited that way. Many are. I mean, he was brilliant in his time, but not all of them are. And so we have 4 that we are going to look at very briefly tonight. And this will be wrapping up our presentation with takeaways and a couple of closing questions. So the 4 that we're going to look at tonight are incomplete dominance, codominance, multiple allele, and X-linked or Y-linked. So these are non-Mendelian. These are, we discovered later that everything's not just dominant or recessive. In incomplete dominance, we're going to have a blend of traits. So instead of a red flower crossing with a white flower and getting multiple versions of either white or red flowers, we end up with pink flowers. In codominance, just like coworkers, both traits show up. If anybody has raised chickens, this is most obvious in what is known as a checkered chicken. And it's, if you look at the feathers really closely, they have a checked pattern. It's not, there's no gray, it's either black or white pattern that's there. Our next one, multiple alleles. This is when more than a single allele pair determines what's expressed. So it's not just one pair of shoes, so to speak, if we stick with my little analogy there, it would be several pairs of shoes that determine what's expressed. The prime example of this is blood typing. Blood typing, the type A or AB or B reside on an I chromosome, or on an I gene, not chromosome, on an I gene. All right, our final one. Our final one is what they sometimes call sex-linked. It's when the trait is attached to the X chromosome or the Y chromosome. Remember that out of our 23 pairs of chromosomes, 22 of them indicate just who we are, but the 23rd pair indicate whether you're male or female. If you have two X's, you're female. If you have an X and a Y, you're male. Well, some traits rest on those chromosomes. Those genes hold those traits. And so that impacts then male versus female inheritance. A prime example of this is color blindness. Color blindness sits on the X chromosome. And so since men get an X and a Y and they only get one X, if they receive the recessive trait, then they're colorblind. So if you can see the number in that screen, then you're not colorblind. But if you can't see it, you may be colorblind. And that's an example of a non-Mendelian inherited sex-linked trait. All right, as promised, we're wrapping up with some key takeaways and a few questions. So the key takeaways from the laws of inheritance. First, start with genotype versus phenotype. Genotype is the combination of alleles, the capital F, lowercase f, capital B, and so on. Phenotype is the observable trait that the gene determines. If it's a Mendelian inheritance, a single dominant allele is going to mask that recessive allele. We can use Punnett squares to determine the probability of inheriting certain traits. Most traits, though, are not inherited so easily. That's known as autosomally dominant or recessive, where the dominant completely overpowers the recessive, as in Mendelian inheritance. Most traits are non-Mendelian, and they might exhibit incomplete inheritance, which is that blend of traits, the pink flower from a red or a white. Or they're codominant, where both traits are showing up, as in our checkered chicken example. Or they're multiple allele, where it takes more than one allele pair to determine the trait, as in blood typing. Or they're sex-linked, where the trait is on either the X or the Y chromosome. All right, we're ending with 3 questions, just like we have all the other segments. Our first question, we're waiting on that delay. Once it pops up, we'll go over it. Okay, which of the following best describes the difference between genotype and phenotype? A, genotype refers to the observable characteristics, while phenotype refers to the genetic makeup. B, genotype is the genetic makeup of an organism, while phenotype refers to the observable characteristics. C, genotype and phenotype are both related to an organism's observable traits. Or D, genotype refers to inherited traits, while phenotype refers to acquired traits. You've got 45 seconds. I got answers already. You guys are so brilliant. Okay, hopefully you chose B for this one. A just had it complete opposite. So remember, genotype, genetic, phenotype, physical. C, simply incorrect. They're not both observable traits. And D, they're both inherited traits. They're both referencing inherited traits. Acquired traits are something that you get in your lifetime, like you learn how to play the piano. That would be an acquired trait, and that's not something you can pass to your offspring. All right, we got two more. Our next one asks us, in Mendelian genetics, why do the genotypes big B, and big B, little b result in the same phenotype? A, because in Mendelian genetics, it only takes one dominant allele to mask the effect of the other. B, because big B, little b is recessive and doesn't influence the phenotype when paired with big B, big B. C, because both big B, and big B, little b contain only dominant alleles. Or D, because the phenotype is determined solely by environmental factors, not genotype. You've got 45 seconds. I got people answering already. You guys rock. Okay, for those of you that chose A, you are correct. Let's break down why the others are incorrect. So for B, big B, little b is not recessive. That's heterozygous. So that makes that incorrect right out of the gate. For C, it says because both big B, and big B, little b contain only dominant alleles. That's not true. Big B, little b is not a dominant allele. Or D, environmental factors. No, that was just silly. That's not in there. So I did just answer why not C. We've got that little b in there. That little b messes that up and it makes that an incorrect answer. All right, our final one of the night, folks. It's going to ask us what, I'm waiting patiently for the delay here. What does a Punnett square determine? A, the exact physical appearance of an offspring. B, the probability of different genotypes and phenotypes in the offspring from a genetic cross. C, the total number of offspring produced by a genetic cross. Or D, the dominance or recessiveness of a trait based on environmental factors. You've got 45 seconds. Okay, hopefully you chose B. It's identifying probabilities, and I think all of you did. It's certainly not going to tell us the exact physical appearance of an offspring. It can't tell us the total number of offspring, and it can't tell us anything about environmental factors. All right, folks. So I have just a few things to go over with you, and I'm going to then attach the slides from tonight's presentation. So just bear with me. One thing I want to draw to your attention, this was at the beginning, but I do know we had a few people that trickled in later, just that we have many, many other things that we offer from Archer Review for your TEAS prep. And I went over these at the beginning, but please know that when you get their slides, the QR codes are in there. So we have our free live webinars that cover all subjects. We have our free TEAS question bank. We have the on-demand videos like I talked about earlier, where we would do that massive 16 square Punnett square if you so chose. We also offer tutoring, so you want to keep that in mind as well. And then finally, I want to just put a shout out for our tutors. I'm waiting. It seems like the lag is really taking its time right now. We have multiple tutors. We have Jasmine. She is responsible for science and math. Tammy does the English and reading. Brandon does the math. And then Amber and I are both science. So Amber was the one who did, if you saw the first one, she did your anatomy and physiology. I am obviously doing your biology, and I will be doing scientific reasoning as well, probably December-ish. And Jasmine is the one who does chemistry. So I'll let you see their faces real quick, and then we finally have a QR code where you can give me some feedback about tonight. What was good? What could be improved? It was my very first live webinar, so I'm sure I have plenty of areas for improvement. Please do follow that QR code and give me some feedback. I did just read that someone said I was awesome and they learned a lot and understood everything. Thank you. That means so much to me. I can't express that enough. I'm going to get this QR code up. It has really taken a little time here. And while this QR code is up where you're giving feedback, I am going to pull those PDF slides for you. Like I said, I did learn that I have access, I can access and give those to you on all of our streams and restreamings. So bear with me here. Oh, you guys are so sweet. You're giving me such wonderful feedback in the chat. Thank you. That means so much to me. Bear with me, that QR code's coming. It is so delayed now. It wasn't quite, I didn't feel like it was so delayed all night, but it's really taken its time right now. There we are. There's the tutors like I talked about. And I'm working on getting you your slides. There's that QR code. Yay, that makes me very, very happy. Please give some feedback. I would love to learn and grow and be a better presenter for you. All right, folks. So now you've got your QR code. Zoom, I am sending out the link to the slides there. And those of you that signed up ahead of time with Zoom, we do have your contact information. If you struggle with opening these or have any difficulty, obviously let us know. Same thing with Facebook and YouTube. I just now sent the PDF, and you can always reach out to us if you have difficulty opening it. I am happy to guide you and get you the PDF. Okay. Facebook is asking, how do you download? And that is a great question. If you're on your cell phone, I think that's difficulty, if I remember correctly. You may have to be on a device, like a laptop or a computer for that. Yeah, I'm pretty sure that's not something that's easily downloaded on all devices. Okay, I'm gonna leave this QR code up for just a couple more minutes. Please do give some feedback. I appreciate it. And thank you guys for sticking with me. It was 2 hours and 21 minutes of a whole lot. So thank you, thank you. You guys have a lovely evening. I'm gonna leave this QR code up for just a little bit. All right, folks, make sure you get that feedback out there with that QR code. Have a fantastic night, and I'm sure I will see you in the future for scientific reasoning, and I'll do other biology webinars in the future. Best of luck to you.